Alright, so let's have a look at how bringing glucose into the cell can actually affect the amount of ketone bodies we're making. So to do this, we're going to consider both healthy and diabetic individuals. So in a normal individual, the glucose in the blood can be brought into the cell via our specific glucose transporters, and this is facilitated by insulin. So remember, insulin is the hormone that brings glucose from the blood into the cell. So once the glucose is inside the cell, using glycolysis, that 10-step process, we can convert the glucose into pyruvate. So the pyruvate can then be converted to oxaloacetate using an enzyme called pyruvate carboxylase. So we can't actually make oxaloacetate unless we've already done glycolysis. So that is key to this process. So that oxaloacetate is actually needed for the acetyl-CoA to be used in the citric acid cycle, because the first step of the citric acid cycle has acetyl-CoA combining with oxaloacetate to make citrate. So that will start the whole process. And a very small amount of the acetyl-CoA is actually going to be used for ketogenesis. Most of it will be used in the citric acid cycle. So remember, ketogenesis is the process where we're making ketone bodies. So there's going to be a small amount of ketone bodies made. And the place we're getting the acetyl-CoA from, it's going to be predominantly from our beta oxidation. We can also get some of it from the conversion of pyruvate to acetyl-CoA, but we've been mostly talking about beta oxidation today, so let's just consider it coming from beta oxidation. So that's what happens normally. However, if you're an untreated diabetic, things are vastly different. So if you're an untreated diabetic and you've taken in a carbohydrate-rich meal, you've got glucose in your blood. However, the insulin that diabetics have may be faulty, or they may not just, the cells may not just be responding to it. So this is problems we have in diabetic individuals. So basically, because there's something wrong with insulin or insulin recognition processes, the glucose does not enter the cell. So that's why we've got this cross here. So faulty insulin means that the glucose does not enter the cell. If the glucose does not enter the cell, then there's no glycolysis, so no conversion of glucose to pyruvate. And because there was no glucose converted to pyruvate, we then don't have any pyruvate to make oxaloacetate. And without oxaloacetate, acetyl-CoA cannot enter the citric acid cycle, because oxaloacetate and acetyl-CoA combine together to make citrate in the first step. However, what we can use the acetyl-CoA for is ketogenesis. We don't need oxaloacetate to do ketogenesis. So that's why we've got such a large arrow here, because the acetyl-CoA is going to be used to make ketone bodies and not used for the citric acid cycle. So it's basically all funneled into the ketogenesis pathway, and this means we're actually going to get an overaccumulation of ketone bodies. So remember, we get our acetyl-CoA from the beta oxidation of our fatty acids, and if we don't have oxaloacetate from carbohydrate digestion, then we funnel all this acetyl-CoA into ketogenesis to make ketone bodies, and this is what leads us to have an overaccumulation of ketone bodies in the cell, and then they'll actually be moved out into the blood as well. So we increase the amount of ketone bodies in the blood. So basically, we can see this summary of what's happening here in these 2 points. So glucose is not brought into the cell due to faulty insulin, and that means that all downstream reactions that rely on glucose are also blocked. And the result of these blockages is that the acetyl-CoA from beta oxidation is actually used to convert into ketone bodies using the process called ketogenesis. So what happens when we do ketogenesis to this extent where we're funneling all the acetyl-CoA into ketogenesis. So if there's no oxaloacetate for the acetyl-CoA to feed into the citric acid cycle, we saw that the acetyl-CoA is instead used to make ketone bodies using ketogenesis. And the accumulation of too many ketone bodies in the blood actually can cause health problems. And one of these is called ketosis. And I showed you on the previous slides that diabetics are actually more at risk of producing lots of ketone bodies, meaning they're more at risk of ketosis, as they are actually have impaired carbohydrate nutrition, which means they don't have pyruvate to convert into oxaloacetate to allow the acetyl-CoA from beta oxidation to actually be used in the citric acid cycle. So when we don't have enough oxaloacetate being produced, the acetyl-CoA is used to make ketone bodies. And this is what we saw in our untreated diabetic, where they couldn't start the citric acid cycle due to a very low amount of oxaloacetate. So the acetyl-CoA was used to make ketone bodies instead in a high amount. So what is ketosis? So normally we have very low levels of ketone bodies in the blood, but if we're in a state of ketosis, where ketone bodies are made more readily, there's actually a 50 to 100 times higher concentration of ketone bodies in the blood. So there's a few other definitions we can use here. So ketonemia means excess accumulation of ketone bodies in the blood, and ketonuria means we've got so much accumulation of ketone bodies that it actually will the ketone bodies will actually start being excreted in the urine. So if there's more than 70 milligrams of ketone bodies per 100 ml, then you can actually see the ketone bodies being excreted in the urine. So ketosis is the general term we use to describe the accumulation of ketone bodies in the blood and the urine. And you might also be able to smell the presence of acetone on the person's breath as well, because remember, acetone is exhaled. So ketosis is harmless in the majority of people who are just on like a low carbohydrate diet or are fasting, but if it goes on for a longer period of time or is a bit more extreme, then we can get some symptoms, including headache, dry mouth, and the presence of this acetone breath. However, ketosis can actually turn into what we call ketoacidosis. So this is where we actually have so many ketone bodies in the blood that they actually begin to decrease the pH of the blood. So by lowering the pH of the blood, it actually makes it more difficult to carry oxygen through the blood. And it is also increasing the amount of urine that's being excreted, so dehydration is occurring too. So if left untreated, the ketoacidosis can actually cause coma and death. So the difference between ketosis and ketoacidosis is basically the extent of ketone bodies in the blood. In ketosis, you're going to have an accumulation, but in ketoacidosis, you have a huge number of ketones in the blood, and this will have happened over a long period of time, and then we start getting these negative consequences of having the low pH of the blood, which is caused by the hugely elevated blood ketone levels, ultimately resulting in coma and death if left untreated. So it's pretty serious in terms of what can happen if you completely shut down your carbohydrate metabolism and are relying on ketone bodies instead of glucose to feed some of your tissues. Remember, some tissues actually can't derive energy from lipids. Things like the brain actually can only use glucose and ketone bodies as fuels. So by having these very low carbohydrate diets or completely cutting it out, you open yourself up to dangers such as ketosis and, in extreme situations, ketoacidosis. So you can see how this fits in with our lipid metabolism picture. So we started talking at the start about our lipid digestion, where we are bringing in our triacylglycerols into the blood, and we can then use these in lipolysis to break down into fatty acids and glycerol. So the glycerol can feed into glycolysis and gluconeogenesis by being converted to dihydroxyacetone phosphate, whereas the beta oxidation allows us to make lots of acetyl-CoA from our fatty acids. So normally this acetyl-CoA is delivered into the citric acid cycle, but this is reliant on us having oxaloacetate, which we get from carbohydrate metabolism, specifically converting pyruvate into oxaloacetate. So if we don't have enough of this oxaloacetate, then instead we'll do more ketogenesis, which leads to that overaccumulation of ketone bodies, where we use a lot of the acetyl-CoA to make ketone bodies. So this can lead to firstly ketosis and then eventually to ketoacidosis, which, if left untreated, can lead to coma and death. So what dictates whether we're going to use acetyl-CoA from beta oxidation in aerobic respiration or ketogenesis? So it's basically how readily available oxaloacetate is. So if oxaloacetate is available, then we're able to combine acetyl-CoA with oxaloacetate to start the citric acid cycle. Then we can keep going through those phases in aerobic respiration to make ATP. But what about if there's not enough oxaloacetate? Then what would happen? If there's not sufficient oxaloacetate, then the acetyl-CoA from beta oxidation is instead used to make ketone bodies in the process called ketogenesis. So what is the problem with an overaccumulation of ketone bodies? So too many ketone bodies can actually lead to ketosis and eventually, in extreme cases, to ketoacidosis, which can lower the pH of the blood and cause coma and death. So we can see how by not maintaining an adequate balance between carbohydrate and lipid metabolism, then we're basically eliminating our, well, not eliminating, but we're compromising our ability to gain energy in the form of ATP from our acetyl-CoA from the breakdown of fatty acids via aerobic respiration. So really throwing out this whole balance. So a little bit about our lipid transport. So remember, our lipids are insoluble, so we need to have specific proteins that actually carry these around the body. So for cholesterol, cholesterol actually uses our lipoproteins, which are low density lipoprotein and high density lipoprotein. So LDL is the one that carries the cholesterol away from the liver to other parts of the body. And this is the type of cholesterol that is associated with the formation of atherosclerotic plaques that open up the individual to cardiovascular diseases, which can lead to myocardial infarction, more commonly known as heart attacks. So that's why we call our LDL our bad cholesterol, because it's basically delivering the cholesterol to other parts of the body, whereas the HDL is actually the good cholesterol that will take the cholesterol from other parts of the body back to the liver, where it can be utilized to make fatty acids. So we actually use the liver to make cholesterol and regulate its function. So we need to take cholesterol to other parts of the body so it can be used in other cells, for example, to make cell membranes or specific hormones. But we can also have cholesterol being used to make bile salts, so which are stored in the gallbladder. So we see there's a lot of different roles for our cholesterol, but we're relying on these lipoproteins to carry them around the body because cholesterol itself is insoluble. And if we have too much of the circulating cholesterol, which is our LDL, then we open ourselves up to these cardiovascular diseases. So in this slide, we can see a little bit more detail on some of the health outcomes if we don't have some of these cholesterols in the right levels in the right locations. So a lot of this is repetition from the previous slide, but let's focus on this last part down here. So the liver removes our low density lipoprotein, remember that is our bad cholesterol, from the blood and bringing it into the liver, and it does this by receptor mediated endocytosis. So basically it's using a receptor to bring the LDL into the liver. So we can have certain metabolic diseases or conditions where we actually have a genetic predisposition to an abnormal amount of different types of cholesterol. So there might be too much LDL cholesterol in the body, too much HDL, etc. So we have a condition called familial hypocholesterolemia, where there's actually a defect in this LDL receptor that actually brings the LDL into the liver. So that means you're going to have a higher level of LDL in the body, which is what we call our circulating cholesterol. So this increases your risk of the cardiovascular diseases like strokes and heart attacks. So basically we want to maintain adequate levels of these cholesterols to perform the functions, but we don't want them to be too high, especially our LDL. And we can see here how the genetic condition actually makes us even more predisposed to poor cardiovascular health due to too much of this LDL being present circulating rather than being incorporated into the liver due to the faulty receptor. So we've had a look at a lot of different things today. A lot of it has also related back to other sessions. So if there was a few points you weren't sure of, it might be a good idea to have a look back at other sessions to make it a little bit more clear. So we saw that once we have our triacylglycerols inside the body, then we break them down into our fatty acids and also the glycerol. So we've basically been focusing extensively on our fatty acids because that's where we can do more things with them. So if we have our fatty acids, we can use these to make a lot of acetyl-CoA, and depending on whether we have oxaloacetate available, we'll either use that to start the citric acid cycle, or we can also use the acetyl-CoA to make our ketone bodies. So we've got another session next time looking at our lipid metabolism. So that's where we'll be more focusing on how to actually generate and store triacylglycerols in our adipocyte cells, which make up the adipose tissue. So I'll see you next time for that one.